I'm on the board of trustees of the Zuckerman Mind Brain Behavior Institute. We have 852 scientists working on the brain, and it's Columbia's main scientific initiative. And the institute is sort of the glue that's coordinating the effort, and we believe that that's going to be the arena in which the greatest breakthroughs are going to come. And the reason it's so important to have an institutional setting for science is the voila moment where someone gets a brilliant idea is still going to be relevant. That great insight that comes that makes a big difference now has to be examined, explored, and the foundation for that great insight is usually built on decades and decades of research. And that research is by necessity now conducted with supercomputers, electron microscopes, imaging machines, MRIs, PET scans, and each of these, and then you have the biobanks, the great biobanks, which are collections ranging from the British Biobank, where they have all of the National Health Service records anonymized, and you can use that as a base to get statistical data with the supercomputers, and that's being replicated at Mayo Clinic and many other great institutions. All of that is requiring an institutional setting as the basis for modern science. And the evidence of that is that Alzheimer's discovered the sticky substance amyloid in 1901. He identified and described it. And all the way up until 1989, zero progress was made. And in 1989, they identified the first gene that had relevance to it. And then there was another dry period. And then since 2017 until now, there have been one major breakthrough in science after another in the Alzheimer's arena, facilitated by these huge technological advances in supercomputers and electron microscopes and things like that. The people who are our guests tonight are the people who work with those instruments. So, Anthony Fitzpatrick took me through his laboratory with his $10 million electron microscope as big as this entire house and stories higher. And so we are here to learn about what these great institutions are doing, particularly Columbia, and what the discoveries that they're working on now or have found recently, and where it's going. And then we'll have some questions as well. It'll be the talks will be distributed throughout the evening, so we'll start with a couple of them before the first course, and then we'll go further in between courses. And our first speakers will be the co-directors of the Institute. Daphne Shahami not only is a great Israeli warrior as a sergeant in the Israeli army, but she's a great scientist, and she specializes in memory and the various factors in memory of how you get motivation, how you store thoughts, how you recall thoughts, and all of these building blocks. She and Professor Axel are really integrating all these scientists and trying to get them into teams and to keep track of all the different things that are being done and seeing where you have singular objectives that different experts could work on simultaneously. And so that's a new initiative. And also the Institute is trying to bring more of the science to the bedside faster. So we're working with Columbia Presbyterian in more applied psychiatry and trying to take scientific revelations and make them more quickly applicable to the patients that are being treated. And Daphne is pulling all that together and spearheading that. And in addition to doing her own brilliant lab work. So we're going to turn it over to her, and then her partner in this, originator of the Institute, is Richard Axel, and his work at the Institute and in creating the Institute, I guess you might say, is complementary to his getting the Nobel Prize. And we're all learning from him constantly and proud of him. His specialty has been odor and all the genetic elements of odor and how it leads to memory and how you interact with your environment with the genetics of the odor and how it allows you to function within the environment. And his work has been cutting edge. He continuously comes up with new and brilliant ideas and work, and has motivated our team, the teams of scientists there, to pursue cutting edge science, and we expect the second Nobel Prize for him soon. And so they will deliver our first talks, and Daphne will describe a little bit about the institution, and we'll go from there.
Thank you, Ken, for having us here, first and foremost, at this gorgeous setting. It's just beautiful and a beautiful evening. Hello, everyone. Thank you for choosing to spend your Saturday evening at the end of August with us. As Ken mentioned, our purpose here today is really just to share with you the science that we are most excited about. Ken gave us very specific guidelines. He said, don't make it too simple. Keep it complicated. We want to know what's really happening in the lab. That's right. He said, these are very smart people. So that's the plan. And if it's too much, you can blame Ken. The plan is really to start by telling you a little bit more about the Institute, how it came to be, and what its purpose is, and then to kind of dig into the science, mostly focusing today on memory and on movement and how our brain accomplishes both of those things, and some of the open questions remaining that we're looking to solve in the next 5 to 10 years. Before I give you sort of more detailed information about the Institute, I'm excited to ask my colleague, Richard Axel, to make some remarks. Richard was one of the co-founders of the Mind Brain Behavior Institute, together with Tom Jessel and Eric Kandel, and in partnership with President of Columbia at the time, Lee Bollinger. And they had this crazy idea to do something radically different in terms of building a research institute. And that idea became a reality thanks to Mort Zuckerman, who gave a really transformative gift. And I'm happy to have Mort's nephew, Jamie, here with us this evening as a great supporter of our enterprise. So, Richard, I'm going to pass this on to you, and then I'll tell you more afterwards.
Thank you. First, let me as well thank Ken for graciously inviting us into our home, into your home, to share some thoughts on the future of the Zuckerman Mind Brain Behavior Institute. And what I'm going to tell you very briefly today is that the Institute, which addresses inordinately complex questions of mind, now recognizes a sense of responsibility, a palpable urgency to apply this insight to neurodegenerative disorders. And I will briefly tell you why I think now is an opportune moment. But a few asides. Firstly, Daphne is the director of the Institute. Hi. Secondly, the Institute was founded about 10 years ago, as Daphne already mentioned, with the truly brilliant foresight of two people, Lee Bollinger and Mort Zuckerman. Mort not only brought his mind to the problem, but graciously provided us with the support to allow us to build out the Institute. And over the years, we have benefited enormously from the nurturing of Mort's nephew, Jamie Gertler. Now, the brain is the most mysterious, the most complex, and the most interesting structure in our universe. Emotion, perception, movement, cognition, memory. Consists of so much activity. This is an assumption. Let's think about it in the context of perception that we're supposed to think about. How is it that wavelengths of light in vision, the chemicals of smell and taste, the frequencies of sound in audition can be recognized and represented to allow you to perceive the world simply by the recognition and activity of a group of nerve cells? 100 billion nerve cells in your brain. And nerve cells can only do one thing, fire. And their firing can only vary in two dimensions, time and space. This is truly a magical problem. And it is a problem that we believe will take decades, if not centuries, to untangle. And perhaps the brain will be unable to understand itself. And we can live with unknowing. But as I said earlier, as the Institute has matured, we now feel an increasing responsibility, as I said, a palpable urgency to bring the insights of basic neuroscience to questions of neurodegeneration. Neurodegeneration, and all of us have experienced individuals suffering from one or another form of neurodegeneration. Neurodegeneration is a most insidious illness. It's an illness that strips away from humans the most human qualities: reason, insight, emotional stability. And I believe that we do not have to wait to understand the inordinate complexity of memory, of movement, to begin to take the insights of today's neuroscience to address problems in neurodegeneration. Why? The functions, these higher order cognitive functions of the brain, need not be understood to understand the cell and molecular basis of neurodegeneration. Neurodegeneration, I would argue, is not a disease of complex neural circuits. It's a disease of molecules that destroy these complex neural circuits. And as a consequence, an understanding of the cellular and molecular events that are perturbed in neurodegeneration should be an adequate target for our efforts. For instance, a nerve cell lives for 100 years. It's remarkable. A red cell in your blood for 90 days, a cell in your gut for 3 days. There is something very special about a nerve cell. And what is the problem in neurodegeneration? The neurons die. And so if we can understand why neurons can live 300 times longer than most cells in the body, perhaps that will provide us insight into why they die. And so, again, the Institute, the members of the Institute, here, a number of them this evening, feel that this is an opportune moment for us to make a responsible move to take our insight into the magical properties of higher order cognition and devote a significant energy to an understanding of neurodegeneration. And it really is a pleasure and a privilege for us to have this opportunity to describe to you the nature of our efforts and our future.
Thank you, Richard, and we fully support your decision regarding the rest of the evening. And there's and the rest of the year. There's a long year ahead. I would like to ask you actually to travel back in time with me. I'd like to give you a little bit of context for how we came to be the institute that we are. And I'd like to ask you to travel back with me to 1937 at Columbia University and to the physics department, to the basement where there was a young scientist, an immigrant, a Jewish immigrant, which was remarkable at the time. And he was obsessed with figuring out the properties of atomic nuclei. Does anyone not have a guess? So does anyone know who I'm talking about already? Isidore Rabi. And he applied this obsession to very hard work for a very long time. I actually just learned recently that he ran out of research funds. And the only reason during those critical years that he was able to continue doing his research is that someone else won the Nobel Prize and gave him half the money to keep doing his work. His work led to remarkable discoveries and to tools that enabled us to measure nuclear magnetic resonance, for which he won the Nobel Prize in 1944. I'm sure when he was making this discovery, the last thing he was thinking about was me, several decades ahead, and my colleagues who would be interested in imaging the human brain, or any of us who may have had MRI scan to measure tumors, back injuries, and so forth. But it is this discovery that he made in the 30s that led to the development of magnetic resonance imaging as a diagnostic tool and as a tool that enables us to measure activity in the human brain non-invasively when we are thinking, when we are processing language, when we are making gestures, when we are having thoughts, when we are creating memories, such as memories of this moment in time. It's been transformational to our ability to study the human brain in action. Much of what we know today about human memory and any new surprising results about the uniqueness of human memory have emerged from this tool that dates back to that discovery. It has enabled us to determine, if I were scanning your brain right now, what is the likelihood that you will ultimately remember this moment. I'm not scanning, I don't know, and I won't be testing you. It's also enabled a very surprising and somewhat poetic discovery, which is that when we put people in the scanner and ask them to retrieve memories of the past, we see a pattern of activity in the brain that is rather unique, but that is nearly identical to what we see when we put people in the scanner and ask them to imagine an event in the future, which we were just talking about earlier. That leads to all kinds of questions about what memory is and why and how we use these circuits for thought in general. How and why and when does the brain actually distinguish between things that actually happened or things that might have happened or things that might happen another time in the future? Speaking of past and future and time travel, I'd like you to travel with me a little more, a few decades ahead. We're still at Columbia, but we're on a different campus now. We're on the medical campus in 1973. Eric Kandel had arrived at Columbia and founded the Center for Neurobiology and Behavior. Eric was obsessed with a very different kind of problem at face value. He was obsessed with figuring out how cells, how brains created memories. What was it in the biology of the brain that changed when you created a memory? Eric was another immigrant, in fact, a Holocaust survivor. And he thought that the psychiatrists he had trained with in studying psychiatry were approaching this problem all wrong. That Freud and psychoanalysis were not going to lead us to understand what it was biologically that was happening, and that it was that biological understanding that would help us figure out both the mysteries of the human experience, but also how to treat and understand it at the most fundamental level. So Eric had another radical idea, again taking a departure from what his field was doing, and looked for an organism with the absolute simplest neural circuit that he could find that could still be measured in terms of its neural activity, its electrical pulses, and where there was still enough of a behavior to show that memories were formed. And as many of you probably know, he turned to the sea slug, the aplysia, and performed groundbreaking work that would result in the Nobel Prize in 2000 for understanding some basics of neural transmission and essentially how a memory leads to changes in the synapse and in electrical activity. Work that, again, has had a massive influence, not just on me, but on many of us. Concepts of neuroplasticity that we use all the time now when we talk about programs in education, even in physical therapy, in literature, really emerge from this biological discovery and how it changes how we think about the human mind. I'm telling you these two stories to emphasize that these were driven by basic curiosity and how important curiosity can be for driving discovery and for changing the world. But I'm also telling them to you because I think many of us have reached the conclusion that that is not enough. We cannot just sit around and wait for curiosity to drive discovery and to change the world. It doesn't work as well as we need it to, and we can see many problems, pressing problems in society that we know will end up having scientific solutions, and we don't have the solutions that we need right now. One of the reasons it's so difficult to just rely on curiosity-driven research is in part because it's incredibly slow. And it's incredibly slow in part because of the structure of academic science, which has always taken place in siloed departments and in departments at siloed universities. And this leads to many obstacles, both in the initial phases of research, where a physicist will be in a separate department from a psychiatrist, will be a separate department from an engineer. And the tools we need to understand the brain now, as Richard suggested, require a collaboration between all these different approaches to understand all levels from the synapse to behavior. It was the recognition of those obstacles that led to the establishment of the Zuckerman Mind-Brain Behavior Institute. And so to come up with a very different model, a very different way of addressing science that we think can be transformative in breaking down some of these barriers, we have the pleasure and really the honor of being one of the largest neuroscience dedicated institutes in the world. We have 54 individual labs, about 850 people, as Ken mentioned, scientists in one place. That number in of itself is remarkable. It's pretty much the size of the Salk Institute in a single institute devoted specifically to neuroscience, the mind and brain. But I really think the unusual thing, and hopefully we can showcase that for you this evening, is not just the size, but the scope. In a single institute, we have people who are studying molecules, circuits, and behavior at all different levels. When we want to understand how memories are formed and how memory is affected by neurodegeneration, we bring together scientists who are working at that problem in many, many different ways. So far, what we've done is shown that environment is truly unique in terms of its ability to promote collaboration. We chart the number of collaborative studies that have taken place in really under the 10 years as we've all been in a single institute. And we can see a massive increase. And I could share with you another time, just from my own research, how being with my colleagues has changed the way I think about the problems we're addressing. We really think this is just the beginning. We are located on Manhattanville campus, the new campus at Columbia University, in a really fantastic building, which if you haven't been to see us there, please come visit, designed by Renzo Piano, really purposely to promote this kind of collaborative environment so that we can take risks and we can talk to each other about the problems we're trying to solve. And really that's kind of the essence of what we're trying to do. And what we're doing now is pivoting, as Richard said, to taking all this, in some sense, all these amazing ingredients and using them to promote and target development specifically about neurodegeneration. And today you'll be hearing about some of those discoveries about memory in the human brain, which is the strange species that I myself study in my lab. We'll then be hearing about cutting edge research that's being done by Gwyneth Card on understanding the computations in the brain that enable decisions related to movement. Another function that we know changes both with aging and with neurodegeneration. And then Anthony Fitzpatrick will tell us about research in his lab that's really getting at sort of the molecular structural basis of some of the changes that we think are happening in neurodegeneration. So for now, I'm going to leave you with a lot of new information. We will have some appetizers. I will come back to tell you a little bit about research on memory, and then we'll hear more from my colleagues. So thank you for bearing with us. Let me just say, both for myself and for my colleagues, we're hoping this will be much more of a conversation than a set of speeches. Please interrupt, bombard us with questions. We're here to hear both the questions on your mind, try to answer them. And really, I think our hope in all of these kinds of situations is to teach you something about what we do, but also for us to learn from you and to think differently about the questions that we're already asking. So thank you again.
Gwyneth is examining, I don't even know how to pronounce it, what, Drosophila or some Drosophila. She is examining the Drosophila flies with the idea of learning how we tend to get away. And so I think that not only will we learn the substance of these scientific responses, but how it translates to knowledge up the chain of complexity. Please, Gwyneth.
Let me just start by echoing Daphne and Richard's sentiments about how grateful we are that you all came out to listen to some science this evening. It's fantastic to see so many faces about to be subjected to some information about flies. So I am going to tell you about my curiosity-driven research. but I think it couldn't have been put in a better context that one of the reasons that I am at ZI, which I have only come to in the last 2 years, is that it was the place that I really wanted to do this curiosity-driven research, because the context and the goals of the Institute in how to shape this research into a deeper understanding of our own brains, or even potential mechanisms for helping with health and disease issues, is something that I couldn't have done in the place I came from, or in a sort of more siloed research institute. So we're really excited to be at the Zuckerman. I am going to tell you something about movement today, and how it is that flies sense their environment and then use that information to decide what to do next. And one of the reasons to study the flies is because we think we can actually answer this question, as opposed to what Richard explained to you so eloquently about our own brains and how impossibly difficult they may be to understand. The fly brain, and I'm talking here about the teeny tiny fruit flies that you might see buzzing around your wine right now. These flies have brains that are the size of a poppy seed. They only have, in contrast to our sort of 100 billion neurons, they only have about 100,000 neurons. If you can imagine that you wanted to go into a brain and understand what every single neuron does, and you're going to do that by listening to every neuron. If you wanted to do that in our brain, you would have to talk to neurons about the number, if you imagine each neuron as a person, it's about the population of 12 Earths. That's a lot of people to interview. Now, you take the fly brain, you only have to go and interview the population of Anchorage, Alaska. This seems much more approachable, and it attracts me because I think that this is something that could actually be done within my lifetime. And I sort of am into the instant gratification. I really want to know how this brain works. Okay, so one of the first things that attracted me to science was movement. It started for me in high school. I was in my AP psychology class, not paying attention, gazing out the window on the lawn, and I'm watching these squirrels hop around on the lawn. And I'm very sleep deprived, and I'm also taking BC calculus, and I'm thinking the squirrels are moving in parabolas. And I became so drawn to this idea that our movement is shaped by the physics of our world. And what our brains need to do is they need to understand something about that in order to select what's the most efficient movement to do at the right time, how to orchestrate these sequences. You know, if you go to master a tennis serve, right, there's a very sort of efficient motion that you're developing to put the most power and spin on that ball. How is it that our brains are learning that and sequencing that? And so I took this interest with me initially into to college, to undergraduate research opportunity, where we were studying biomechanics. And I thought, okay, I'm going to finally understand how movement is generated. And this is biomechanics. We were studying wallabies and kangaroos hopping. I was telling some of you I had the privilege, I got to go to Australia with my lab, literally in the outback of Australia, chasing kangaroos over force plates to see how they generate this movement. I can tell you all about the efficiency of kangaroo tendons if you want. But it became clear to me that really the answer to how our bodies move is not in the body. That's part of it, but really the commands come from our brain. And so this is what brought me into neuroscience, and actually what brought me into flies. So I told you a little bit about why to study flies, because they're compact and we think we can understand them in our lifetime. But there are many other reasons, and most of those are tools. Now, we've heard many stories this evening about the sort of inspirations for our work going back to those before us who came from Columbia. And the story in my field is no different. Of course, Thomas Hunt Morgan was famous for starting at Columbia University, a place he called the Fly Room. And he's the one who initially chose the species Drosophila melanogaster to bring it into the lab to make it an organism to study. He was interested in genes and the basis of heredity. He eventually won the Nobel Prize for that work in 1933. But in addition to sort of initiating the modern genetic era, he really was the one who made Drosophila, this fruit fly, an organism we could study, because now that we saw how its genes worked, we could start to manipulate those genes for scientific research purposes to affect the animal. And fast forward it to the last decade, we now have tools in the fly where we can manipulate their genes to target specific brain cells, individual neurons, which means I can go into my fly and I can turn a single neuron on, I can turn a single neuron off, or I can express fluorescent proteins in that neuron so that I can watch it blink on and off when the fly is behaving. And these are the components that you need to go in and be able to understand a system as complex as the brain. Now, there's one other new cutting edge fly technology that has really made, I would argue, the fly one of the forefront research organisms for modern neuroscience. And this has just happened in the last five years or so. And that is something we call the connectome. Okay, what is the connectome? It sounds a lot like a genome. And in fact, that's what it's inspired from. The idea is that you can take the fly and you can understand these 100,000 neurons in completeness. And to do that, you want to understand how they're connected to each other. How does neuron A talk to neuron B? How do you go from a photoreceptor cell that senses light all the way to a motor neuron that's going to drive your muscle? We need to know the pathways. We need to know the links in the chain of how you get from one to the other. And having a roadmap for that is key. Now, of course, this kind of roadmap is hard to make in brains as complicated as our own, or even other research organisms such as mice, which have many, many neurons. But in the fly, we've been able to take electron microscopy techniques. This is basically a kind of microscopy, not quite as fancy as Anthony's, but still gets you really, really good resolution, subcellular resolution. And researchers have made thousands and thousands of images of little parts of the fly brain capturing these neurons and their shapes and their interconnections. And then teams of dozens and dozens of poor misguided students have spent a long time tracing those neurons through these pictures until we now have a whole map. We know the fruit fly's brain in its completeness. And that is what the fruit fly has to offer us now, this idea of completeness. How can we understand a whole brain from soup to nuts? And this has happened at exactly the right time, because as people have looked at other brains, our brains, mouse brains, and we've been able to look at more and more neurons at a time, it's become apparent that there's almost no phenomenon that is localized to a single part of our brain. Sure, parts of our brain are specialized for different kinds of functions. But if you ask, how did you reach out your arm to pick up your wine glass? Probably almost all of your brain lit up when you did that. So we really can't be isolated anymore and just studying one part of the brain at a time if we want to understand how the whole thing works and if we want to understand where we need to intercede in cases where it goes wrong. Okay, so now we have this connectome. What I want to tell you today is basically a story of how we've used a particular behavior that Ken alluded to, how flies escape from predators. This is an innate response that flies have, and how we've tried to trace this behavior all the way through from the sensory end to the motor end. Okay, so we took a situation in which we had our fruit flies and we put them in a cage with damselflies, which are basically small dragonflies that we caught out in a pond near our lab. And we filmed with high-speed video recording at 6,000 frames per second. What do these two insects do when they interact? And sure enough, you have a Drosophila fruit fly sitting on the wall of our cage. You'll have a damselfly coming. It hones in on the fly and it darts in as fast as it can to grab it. What does the fly see? Well, it looks like a baseball being thrown at your head, basically. This is what we call a looming stimulus. It's an expansion of a shadow on your eye that occurs really rapidly. This is a salient stimulus, not just for the fly trying to avoid predators, but of course for us as well. If you ever had a baseball thrown at your head, you know you have to react quickly, and you do so innately. Okay, well, here's another important thing to understand about this behavior for the fly. When the fly sees this kind of predator attack, it doesn't do a sort of knee-jerk response, nor do we. We're sitting there and we're thinking very rapidly, but we're thinking, do I duck? Do I try to catch the ball? Do I hide behind somebody else? And the fly is doing the same thing. It has many strategies to avoid being eaten. It could hunker down. It could take off. Flies are lucky that they could fly. It could simply try to run away or hide behind something. And this is the kind of decision making about what action to take that we wanted to understand. How does this happen? Okay, so we turn to our connectome to try to figure out how is information flowing from the fly's eye to the fly's muscles. And the first thing that we see when we look at the connectome is that in this flow through the fly's brain, there are some bottlenecks. I was trying to think of a good analogy, since I don't have any pictures here, for how you can imagine these bottlenecks of the fly brain. And here's what I came up with. Let's imagine that the fly's central brain, we call it, this is where most of the thought processing happens, is the island of Manhattan. And we're going to say that the fly's equivalent of its retina, we call it the optic lobe in the fly, is New Jersey. And the place where the motor neurons are, there's the spinal cord equivalent of the fly, we call it the ventral nerve cord, that's going to be Long Island. Now, if you imagine that you want to drive from New Jersey through Manhattan to Long Island, you have to do what? You have to go over some bridges or through some tunnels, right? And as we all know, that's where the traffic gets backed up, right? Those are the bottlenecks in the flow from one location to the other. And the same thing is exactly true in the fly's brain. It has a part right behind its eye called the optic lobe that's doing some immediate computations on the light that's coming in. But then that information has to be passed on to Manhattan, to the central brain, and it goes through this constriction of just a few channels, just a few neurons. In fact, there's only about 100 different cell types, we call them, types of neurons, that carry this information, compared to sort of the tens of thousands on either side of this bottleneck. And then similarly, if the central brain now wants to give some commands down into Long Island, our center of our motor neurons, it also has to go through a bottleneck. And in the fly, because we have the connectome, we know every single neuron that carries these motor commands from the central brain down into its nerve cord. There's only 1,300 of these neurons. Okay, so what does the fly see then when it sees this looming stimulus, this predator coming at it? Well, we can go to this first bottleneck, these bridges between New Jersey and Manhattan, between the optic lobe and the central brain, and we can just say for each single one of these, how is it responding to different visual stimuli? What exactly is being represented? We can go through and comprehensively survey everything that the fly is processing about that visual stimulus. The specific experiment we did is that we took flies for whom we've manipulated their genes so that we can express a fluorescent marker that blinks on and off when those neurons are active. But we've now used our genetic tools to restrict that to just one of these channels at a time, right? One of these cell types at a time. And we can do an experiment that sounds very simple, go through and see which each of these cell types responds to, but is impossibly hard in organisms that are not the fly with these tools. And okay, so theoretically, there's two ideas for how we might see the world. One is that you might have neurons that are distributed amongst the statistical properties of the world. So sort of abstract, a little bit of movement over here, a little bit of movement over there, and they sort of statistically cover all the different kinds of movement that might be possible. That's one option. Option two, theoretically, is, okay, well, we might have neurons in our brain that are exquisitely tuned for relevant motions that we see all the time. For that predator coming at you, you might have a neuron dedicated to that. You might have one for something that's going across your path, or for some other kinds of motion if you're, say, falling out of a tree, upward motion. Okay, does anyone want to venture a guess as to what is going on in the fly's brain? Is it sort of a more statistical, abstract representation of the brain, or do we have more ecologically representative neurons? Option B. Good guess. Good guess. I'll tell you the answer. It's the same answer that's almost always the answer in biology. Both. You are, however, right in that one of the salient divisions is in terms of the ecological relevance. So the neurons we surveyed, half of them responded to sort of small objects moving around in the fly's visual space, but half of these neurons that we surveyed responded to some kind of looming motion. This is a lot of the brain dedicated to processing looming signals, and this was our first mystery. Is it simply that flies are basically the snack food of the animal kingdom and they get attacked all the time, and so they want to devote a lot of their brain to avoiding this? Or is there something deeper going on? And again, using the tools in the fly, we were able to assess this. So long story short, for the purposes of this evening, we were able to go in and very specifically examine the activity in these different types of looming responsive neurons while the fly was responding to sort of movies of predator-like attacks. And what we found was that, in fact, these looming neurons that from our first pass looked like they were sort of all similar were actually very different. They responded to very different specific computations about looming. In particular, we found one cell type that responds with an exact profile of the speed of the looming expansion. This is telling the fly how fast this object is coming at it. And another whose response was proportional exactly to the size of the looming object. So this is telling the fly how close is this object to you. And this was exciting to us because, in fact, those are computations, those particular looming computations, that have been seen in other animals' brains as well. In locusts, they've seen these actually in pigeons. Pigeons are very sensitive to looming motion, and in mice. So it's very likely that this exact computation about the world, about the motion in the world that's happening in the fly brain is happening in your mammalian brain as well. Okay, but what can we do in the fly? We can go farther, right? We're only partway through our journey. We've only gone through the sensory system. In other animals, we don't know what happens next. Okay, so we can make these computations, but what does the animal actually do? What does the brain actually do with that information to decide on a course of action? Well, in the fly, we turned to the connectome. We looked up those neurons in our map, and we saw who they provided input to. And lo and behold, one of the neurons that they provided input to was what we call a descending neuron. A descending neuron is exactly one of these bridges between Manhattan and Long Island. It's a neuron that gets information from the central brain and then provides output to the fly's nerve cord. And this particular descending neuron was getting input from both these two flavors of looming cell, from the size and the speed neurons. And what neurons do is they tend to integrate, basically sum their inputs. So you can imagine this descending neuron, this downstream cell, was basically computing when something was fast and large coming at the fly's head. And again, turning to our genetic tools in the fly, we can then say, okay, that's great. What does this cell do? Okay, so now we turn to a different kind of tool. We can take our genetic manipulation ability and we can express in this downstream neuron a channel that it doesn't usually have. This is a channel that you put into the cell that is light activated. So when we turn on a red light, it opens. And what that does is it sparks activity in that neuron. It turns it on. And this lets us see what activating that neuron would do to the fly. So we do this in front of our 6,000 frames per second video cameras. And lo and behold, the fly jumps into the air. So we found a neuron that triggers the fly jumping into the air. So we've now completed in some ways our journey all the way from the visual system of the fly, making certain computations about the world, and then connecting that to what the neuron actually does. But there's another part of this story, because this is where things get a little complex. This descending neuron is, of course, not the only neuron that's getting this kind of looming information, and taking off is not the only thing the fly can do. In fact, the fly has more than one kind of takeoff. It does two kinds of takeoff. One, the kind activated by this descending neuron I just told you about, where the fly just jumps into the air, throwing caution to the wind. It tumbles in the air. It's completely uncoordinated, but it gets off the ground as fast as possible. Now, if the fly has a little bit more time, what it does is it actually puts its wings up so that it can do a very elegant downstroke at the same time that it jumps off the air. Now it has a controlled trajectory in flight, but it takes it 8 milliseconds longer. Side note, we have used our genetic tools to silence this descending neuron that leads to the faster takeoff, put those mutant flies in a cage with the predator. They get eaten more. So this is a real life or death decision. Should the fly use the faster takeoff, but then be kind of uncontrolled in the air and have to spend a lot of energy recovering, or should it actually use its more controlled takeoff? How does this come about? And so there's this one descending neuron I told you about that leads to the fast takeoff, and there's a whole population of other descending neurons that coordinate this longer takeoff. Now, what I'm going to tell you, how the way it works is actually the way that we think a lot of decision making, especially about these kind of rapid behaviors, happens in the brain. And it's not that complex, but you have to twist your mind a little bit, because I think the way that people sort of intuitively think about how decisions are made is you think, okay, I kind of accumulate some thoughts about an action or a pathway, and then I decide. And you might imagine that what that looks like in your brain is like, ding, the light bulb's going off, a certain neuron is activated, that's your decision neuron, and then that decision neuron, right, can coordinate a path of behavior. But in fact, a lot of decisions are emergent properties, right? And so this is where Richard was alluding to a little bit, well, maybe our brains can't comprehend our own brains, because it's a little hard for us to understand emergent properties, but this is how it works in the fly. Okay, you have your descending neuron pathway leading to the fast takeoff. You have your other pathways leading to the slow takeoff. We know the fast takeoff pathway gets input from these two looming inputs, right? Size and speed. So if you have something fast and close, then you trigger this descending neuron to the fast takeoff. It fires a spike, actually a single action potential, and that drives the fast takeoff. But if you have a predator that's approaching a little bit slower, right now you don't activate both of those looming channels at the same time. You actually only activate one. You might not activate the speed channel, just the size channel as it gets closer. And that means that you don't trigger this one fast descending neuron, or at least not yet. You actually trigger the other pathways leading to the slower takeoff. And so those ones will then enact the slower takeoff. Which is to say, there's no decision neuron. There's no node in this network that represents the whole decision. It's actually a race between parallel pathways driving competing behaviors that get input from some different combinations of these overlapping looming detectors. And how that plays out in the network over time is actually what leads to the fly's particular action. So we think that this kind of emergent properties, emergent motion decision might be a lot of the ways that these activities are guided. Let me leave you with one more story to give a little bit of indication of where we think this thing can go in the future. So first of all, let me note that even though we are studying the humble fly, there are some analogies, right, between the fly's brain and our own brain in terms of type of computations that are done. We can go into the fly's brain and we can understand in detail these pathways in ways that we can't on our own. We would like to continue this kind of logic in order to understand the whole fly brain, hopefully paving the way to understand our own brains. For example, we already understand these points of bottlenecks. These are points of potential intervention. If you wanted to, for example, create neural prostheses in order to help people move when that function has been disrupted, you might want to put those prostheses at one of these bottlenecks. And you might want to really understand the code, which we can in the fly, in order to guide movement. But one of the questions this raises is about our own free will and our own ability to truly drive our own actions, right? So let's take this fly. I just told you that I can know every single neuron. I know all the pathways, and I'm now starting to know exactly what each neuron computes. And I talked you through a sequence from sensory processing to motor output. Does this mean that I can predict what the fly is doing? Shouldn't I know, right? If I give it a particular visual stimulus and I know how the brain processes that visual stimulus and then how those neurons are hooked up to all of the different motor neurons, shouldn't I know exactly what the fly is going to do? We put this to the test. Over the decade that my lab has been functioning, we have gathered tens of thousands of videos, high-speed videos, of flies responding to looming stimuli and taking off. And so we simply asked, okay, if we look at the first part of the video before a stimulus starts, can we predict the choice the fly is going to make at the end, whether it does a fast takeoff, a slow takeoff, or some other kinds of action? The answer, absolutely not. Even in an animal where we think we can understand everything, and which is vastly simpler than ourselves, we couldn't predict what the fly was going to do. And we've started to make inroads on why. And here's where we think we're starting to touch the magic of the brain in this small species. So we could take our flies. We usually just test one fly at a time. So we put our fly on our platform, show it our looming stimulus, record that, fly flies off, next fly. So we said, okay, well, maybe the reason we can't predict the behavior is because flies have personalities. Maybe individual flies just have a preference. A fly A always likes to do the fast takeoff, and fly B just likes to run away. So we tested that. We then had a postdoc who laboriously had to recapture our flies, put them back in our system so we could test them again. And what we found was that flies that we showed looming stimuli to again and again, sure enough, they like to do the same thing. If the fly did the fast takeoff the first time, it also liked to do the fast takeoff the second time. So we thought, aha, flies have personalities. This is great. But then we're scientists, so we wanted to just double check and make sure. So we did yet one more experiment where we now took those flies that we had tested multiple times, we put them in their home vial for the night, and we brought them back the next day. And we did the same experiment again, test those flies multiple times. Sure enough, on day 2, again, flies tended to respond to the stimulus the same way on a fly-by-fly basis. But their response had no correlation with their choice the day before. So here's how we're starting to understand how this works. We think that flies don't necessarily have individual personalities, but more that they have states of mind. And that these states of mind are not always observable in terms of their action or posture or anything we can see in our video. They're really internal states. And that these have a very, very strong influence on their momentary decisions about how to act, which makes their behavior completely unpredictable. And we're starting in the fly because we have this connectome and we can start to trace all these pathways. We're starting to try to figure out where do these brain states come from and how are they represented? And so we can do experiments like use our genetic tools to activate different kinds of neuromodulators. So neuromodulators are things like the dopamine in your brain or the serotonin, or in the case of fly, they have an equivalent of noradrenaline called octopamine. And when we stimulate the octopamine releasing neurons and we release a lot of octopamine in the fly's brain, sure enough, that biases their behavior strongly to do a takeoff. So we think that there are these internal molecules that are strongly influencing our behavior. And now we can actually start to pick apart that part of the mystery in the fly as well. All right, I'm going to end there. I'm happy to take questions or continue the conversation over dinner. But again, thank you so much for coming out to listen to some of our science tonight.
Our final speaker is going to be Anthony Fitzpatrick. He is multifaceted and talented. He's both a molecular chemist and a molecular physicist, a biophysicist, and he uses all those talents to investigate the formation of amyloid, which is that sticky protein that's found on brain cells of Alzheimer's patients and ALS patients and all these brain diseases often manifest themselves with a substance interfering with the neurons, presumably communicating. And he studies the formation of it and what changes it makes in the cells and in the neurons, and what that leads to. And as he once explained to me generously at a lunch, he's going further, that when at certain points of aging of cells, I'm not the expert, he can tell you more about it, but at certain points of aging, the cells, the neurons can explode, and then the amyloid gets spread to other genes and cells and around the body. So he's the man who kind of bridges in the brain what's going wrong, how that's coming about, and presumably, I believe he worked on the Eli Lilly Alzheimer's drug, which has had some success as a consultant. And so this is the closest we're getting. It's the most basic science that is necessary as a building block to get some impact. Maybe not solve the cause, as somebody was saying earlier, but how to interfere with the negative consequences of the results of whatever it's causing. So with that, we have our resident genius, and he'll elaborate all of the things.
Thank you. Thank you very much, Ken. Thank you very much for the kind introduction. Jimmy Zimmerman was asking me at dinner, how did you end up getting into this field? And like many of us here, I'm sure I got waylaid at some point by a very charismatic man called Sir Chris Dobson, who's now dead, unfortunately. But his view was that the only way to really work on neurodegeneration as a kind of class of diseases is to really, instead of looking at it, as Ken alluded to earlier, from the top down, which is what medics have done for over 100 years, starting with Alzheimer in 1901. Really, you should take a bottom-up approach. So Richard Axel mentioned this earlier. One of the things that's clear about both Alzheimer's and other neurodegenerative diseases is that there is something that's starting within cells. So it's starting at a cellular level, and then as Gwyneth very elegantly described earlier, those cells and circuits starts to then kind of have a cascade of an effect, where it's almost like a set of dominoes. So one cell goes bad, that cell is connected to many other cells, they start to go bad, and so it starts to almost in a way spread throughout the brain, and that manifests in the sort of gradual impairment that we see in people with Alzheimer's and other horrible neurodegenerative diseases. And so really what my lab is doing at Zuckerman is to really focus on the subcellular. So Gwyneth had mentioned the connectome, which is how different neurons and other cell types in the brain connect to each other. We are going right into the very heart of single cells and trying to understand what is that first event that actually tips this whole thing off into a kind of cascade. And we've had hints about that for a very long time. Ken mentioned Alzheimer. So one of the things that Alzheimer found was that when he looked at the post-mortem brain of people with the disease that now has become named after him, he found that the neurons were kind of clogged up with very clear globs of kind of gunk. And that sort of showed very clearly that the neurons were filled in a way that healthy neurons from someone who died of a heart attack or something else did not have in their brain. And to Richard's point earlier about, neurons and your other brain cells have to last a lifetime, which can be up to 100 years or more because they don't divide, you have to think that all of the protein molecules that are inside each individual brain cell, they're all performing very important tasks to the maintenance and upkeep of your cell, and that for those to survive 100 years or more, they get recycled all the time, but sometimes those protein molecules, which have each got individual functions, and those functions are defined by their three-dimensional shape, sometimes they don't adopt that shape, and they sometimes start to accumulate in ways that cause this gunking up of the cell. And there are some proteins that are more prone to doing that. We thought that we knew all of them. As you'll hear later, we did not. But the famous ones that Ken mentioned with regard to the Lilly drug and also drugs from Biogen, most of them target a protein molecule called A-beta. And A-beta is an important protein throughout our lives. It does actually have a physiological role, but either for you have a genetic predisposition towards it, or other causes, lifestyle causes, or just plain bad luck, A-beta can start to accumulate in your brain. And then about a decade later, a second protein called tau also starts to accumulate in your brain. And later in life, most of us, I'm 45, I already have some tau aggregates in my brain. These things are filaments that build up inside each neuron. And to give you a kind of idea of scale, because we've already heard mention of electron microscopes, they're about 10,000 times thinner than a human hair. So they're incredibly fine, incredibly thin. And so there has been a general lack of understanding of how they form, what they look like, how they can be kind of slowed from growing further, because until about 6 years ago, we could not resolve their structure with light microscopy. And then in 2017, we published a very seminal paper on the first structure of a tau filament. And that tau filament can now be used to kind of design, based on its three-dimensional structure, better sort of markers for disease, so things that will bind specifically to this filament, so that when you go for a PET scan or a brain scan or MRI, you can in fact understand better where different tau aggregates have built up. And then moreover, can you develop based on that structure, because we really get a three-dimensional structure of this from this fancy technique, cryo-electron microscopy, which is really just a fancy word for the most powerful electron microscope in the world. That's all we have. And we have it at Zuckerman. We actually have 3 others at Columbia. Columbia in, I think, 2018 or 19, got a Nobel Prize for this. Joachim Frank won the Nobel Prize for cryo-EM for work he did over the 80s and 90s. And so it opens up a whole new kind of set of possibilities about understanding not just the structure of normal physiological protein machines, but particularly these kinds of aggregate that accumulate in many neurodegenerative diseases. And what's fascinating about them is that each neurodegenerative disease seems to be associated with the accumulation of 1 or 2 proteins. So we already mentioned A beta and tau. They accumulate in Alzheimer's disease. But alpha-synuclein accumulates in Parkinson's and dementia with Lewy bodies. And then also in 2008, I think, there was a 4th called TDP43 that was found to be in ALS or Lou Gehrig disease, and also other conditions like frontotemporal lobar degeneration. And I think one of the things that we as a lab have found, which was very exciting just 2 years ago, was a 5th protein that forms these kind of tangles. And if you all turn to page 5 of the document in front of you, this is what we put in your pamphlet earlier. You don't have to turn to page 5, but it was really just a very novel finding that we didn't expect. So we find that there was a 5th protein that we had no idea that even existed. It caused a lot of excitement at the time because TDP43 was the last one discovered by a group at Penn, Virginia Lee and John Trojanowski, using biochemical methods. But here, for the first time, because we were able to image at higher resolution than had been done before, we were able to actually identify a totally new protein that forms these aggregates or filaments. And the way we went about it was also pretty novel, because typically with these kinds of cryo-EM structural studies, you know what you're looking for. So you know that you're looking for dopamine receptor or something that is really well defined. But if you're just going into post-mortem tissue from people who had Alzheimer's disease or Parkinson's or FTLD, TDP, you kind of already expect to see either a tau filament or an A beta filament or something of that nature. And so we kept finding this new filament that we had no idea. It didn't match any of the other previous protein folds or structures. And so we had to get down to a resolution where we could actually resolve the individual building blocks or amino acids of the protein filament itself. And by doing that, we effectively were able to almost identify the amino acid sequence directly from the images. And now this represents a new drug target because we found it in many different neurodegenerative diseases. One of the findings that we've had recently, which is why Ken and I were having lunch, is that we actually find that it not only is disrupting, by forming these filaments, one of the central kind of recycling centers in cells. So there's something called the lysosome, which really is kind of almost like the city dump. So whenever we're sleeping at night, that's when a lot of these aggregates are kind of cleared out. And a lot of the time, they get sent directly to this organelle called the lysosome, which is held at a much lower pH value, more acidic than the rest of the cell, and has a lot of enzymes that can chew things up. But this protein, TMEM106B, is actually an integral part of that lysosome. And so by forming these filaments, it no longer can do its physiological role and is turned over to the bad side, the dark side, and is now forming these filaments, thus preventing the lysosome from doing its role But also what we found in recent months, which we're trying to prepare for publication now, is that it also drives tau pathology. So by forming TMEM filaments, you're also getting additional tau filaments, and tau is kind of an extremely common and very important filament that forms in many different diseases, particularly Alzheimer's. So I think that's what we're working on. That's what we're excited about. I was very grateful to both Richard and Tom Jessel for investing in cryo-EM at the Institute. And I will close, I'm open to questions. I will close with a quote, which I'll have to look at, because I'll forget. So there was a very famous quote from Isaac Newton. If I have seen further, 1675 ladder to Robert Hooke, it is by standing on the shoulders of giants. And I would update that to 2024. If I have seen further, it is by using cutting-edge technology supported by particularly the members of the Brain Trust and also all of you for being here. So thank you very much for your attention.
Now, I know it's been a long night, and you've had lots of brilliant people give you lots of information, and the food was good. So if you're willing to stay, and though some of you might have to leave, and feel free to leave, but if you're willing to stay and people have questions, we have the resident geniuses here, so feel free to ask whatever questions you might have. Thank you, Ken. Thank you, Ken.
So I guess as a layperson, we were talking a little bit about this. So when you get into your 60s and 70s and you're concerned about what can you do and what have you found that may help to prevent any type of dementia, etc., what have you found that may help? Because you get on the internet, there's 1,000 different supplements. There, and I have no idea what really might be helpful.
So I will preface this by saying I am not a doctor. I am a biophysics PhD, but I think one of the people that has a really big interest in this area is actually our host, Ken, who illuminated me recently with a couple of things that we talked about over dinner, such as the big jump, the big sort of difference if you do or do not take hormone replacement therapy. Diabetes is a big driver. Always there's some sort of a very strong correlation there. So I think that, and genetics also can play a major role, which there's not much you can do about that. But so, I mean, I think that for the current knowledge from my reading and from what other more sort of MD types and MD PhD types tell me is really just try as best you can to have a healthy lifestyle. I mean, I think beyond that, there isn't really anything yet. I think that the drugs that Ken alluded to from Biogen and Lilly, they both target different types of this A beta filament, different structures of those, which also has a knock-on effect associated with tau accumulation as well as a filament, but they are very far from perfect. The current idea with the drugs is to slow it down to the extent where, if people live 15 or 20 years longer, if you could slow it down for that amount of time, they would not actually develop the disease. They would already have passed from natural causes. And so the idea is to slow it. It's not to reverse it, or you can't really undo. If a neuron has died, it's died. And that was what Ken was mentioning earlier. that one of the ways in which the fibrils can spread through the brain is that neurons die and then their contents are released and then they're taken up by other cells. And that's also part of how we think it spreads throughout the brain. So it's a very unsatisfactory answer, but it kind of reflects a lot of the sort of lack of knowledge in the field at the moment.
Let me pick up a little bit on that with layman's knowledge, but studied. There are several things that we know. No supplement has really been found to make a substantial difference, but lifestyle does make a very substantial difference. And one of the recent findings is that when you start lifestyle changes at any age, rather than there's like a window you miss at 40 or 50 or whatever, you get benefits from that, even if it's reduced. So if you start something at 40, you might get 15 years. If you start something at 60, you might get 9 years. But there is a relative amount you benefit from lifestyle. What have they statistically found by studying the biobanks? And these are on massive studies of like 500,000 people, million people. Number one is nutrition. Having basically, not to get lengthy about it, a Mediterranean type of diet, very low intake of red meat, very low intake, if any, of alcohol, certainly no smoking. Actually, Daphne, it's very funny, but the wine, I went to Sardinia, and there's some special wines in Italy that are high mountain wines. And these are, I'm experimenting with these high mountain wines because they have polyphenols in a great amount in order to let the grapes survive in the high altitudes. And these polyphenols are very good for your brain and your heart. So this is, you're part of an experiment. But in any case, very low intake, very low intake of alcohol, and sleep. 7 to 8 hours of sleep is like critical. Many people don't get that, but it's a very important factor because Anthony made the point. It's during that period that the liposome and all these functions of clearing occur. So if you sleep 4 hours, you're just allowing the dump to accumulate in your cell. So sleep is a really important, and it's only in the NREM portion, the latter portion of your sleep, that this is occurring. And taking sleeping pills and things like that actually interfere with NREM sleep. You sleep, but you don't get the NREM sleep, where that clearance that Anthony was talking about. You want to do a lot of reading, Lauren. Reading is a critical factor, particularly novels. And the reason for that is you have to keep a lot of characters and plot points continuously in your head. And all these different connections are having to be formed to maintain that. So the more complex the material, particularly novels. So go get Tolstoy and go at it. and then staying away from air pollution, not being in polluted environments. And Anthony already mentioned making certain that you're having immediate treatment of physical deficits like diabetes, hypertension, because those interfere with your insulin functioning, and that is allowing the sugar to get into your blood and form new amyloid and new tau and all these substances. So you want to immediately deal with that. And the final thing I'd say is, if you have any hearing deficits, it's critical to deal with them very quickly, because the hearing deficits have lead to the death of the neurons as they go further and further toward your outer ear. The visual neurons fill that space up, so it's a continuous pressure of them to move further and further away, and more of them die, and you get less information coming in because of a hearing deficit, and so it has to. Unfortunately, a very small number of people who have hearing deficits actually treat it, which is startling, but it's true, and that's one of the big causes. That's the largest cause that's can be remediated easily. So those are some ideas that the current literature has elaborated.
What most of us start experiencing in even our 40s and 50s of the everyday forgetfulness that most people are very troubled by, that is not the start of dementia. That is the very natural result of what we consider healthy aging, and it's due not to changes in the structures that are critical for memory itself, but it's due to natural changes in the prefrontal cortex, which leads us to not be able to get the information on demand, which is different from memory loss of the sort we see in dementia. So even though the experience of it might be equally frustrating, it's a different thing. For that, I think what you do for that is enjoy the silver lining, which is that the loss, the kind of relatively early deterioration in healthy aging of the prefrontal cortex also comes with what many people refer to as wisdom, seeing the bigger picture. There's sort of cognitive changes that are very positive, and there are actually some evolutionary theories that suggest that it's part of being, kind of providing additional learning opportunities to the younger generation, so that it's not necessarily bad, but that it's a trade-off of sorts. It's all the prefrontal cortex is also the latest structure to develop. Yeah. So it's not. I'm trying to end on a little bit on a positive note there, but it's not all bad news.
Well, I think I touched on it actually, because when we found this 2 years ago, a lot of people said, "Okay, what's it doing? Like, why is it doing this?" History of tau is about 40 years. History of A beta is a bit longer. History of TDP43 is about 15 years. So this discovery kind of opened up a whole new set of questions for us. Because TMEM106B, a lot of these other proteins like A beta and tau, they're just around. They're kind of there. They do important things in the cell, but they're not localized to one place in the cell. So they do they do multiple jobs. Whereas TMEM106B is integral to this lysosome. So this kind of city dump where everything gets taken to, and it's a source of regularly dealing with a lot of the junk that builds up in our cells, daily life, everything. And so what we've been trying to do for the last 2 years, which we've now finally gotten, we're writing up a paper now about it, is to understand what that TMEM106B is interacting with and what other aberrant processes it's driving. And we find out, surprisingly, that it's actually accelerating tau aggregation, which is a much more kind of commonly accepted pathological target. So, because one of the things about TMEM is you get these fibrils similar to tau. You find them in old people as well, like, with no symptoms. There are just a lot more of them in certain diseases. And so the question is what are they doing? And I think we're going to follow up on that over the next, few years, 5 to 10 years. We will focus largely on the lysosome and its function in disease, because currently, when I was a PhD student, all roads led to mitochondria in these diseases. These days, everyone seems to be focusing on the lysosome, because that kind of ability to turn over junk and get rid of it on a daily basis, if that starts to go wrong, you will start to automatically have an accumulation of junk in your cells. And that can only be bad, not just for the cell, but also for the cells connected to it via circuits and the rest of the brain as well.
And actually, that in an interesting way brings us sort of full circle, right? Because as you're already understanding, there's sort of what Anthony's describing in terms of like the most recent discoveries at the level of the cell. They have an impact on other cells and on circuits and on the whole brain and on behavior. And those are these connections we're trying to make. And so the importance here is being able to follow up on these discoveries really quickly to understand what changes in the cell and how it communicates with other cells. We heard about the connectome. And what does that have to do with changes downstream? Because if there's one thing we really recognize now is that while the originating problems might be at the cellular level, what manifests in behavior has to do with how those cells impact the whole circuit of interconnected structures in the brain. So that's the constant challenge, is to keep both of those perspectives in mind.
I'm on a health panel at Sinai where it's come to my attention that women are 66% of the people that get Alzheimer's. I find that really shocking and painful to hear, because I certainly don't want to get it. And I'm curious, are you guys doing anything, or do you see them doing anything in terms of estrogen in women to understand why that difference is there?
Yeah, thank you for asking that for everyone. We discussed it a little bit later, and the short answer is no, we aren't right now. And as we mentioned, it's really a very, it's not even a new field. It's sort of there are new smatterings of findings that I think are going to lead to the birth of a new discipline. We mentioned, that generally speaking, women's health is understudied. Women's brains, and whether there is such a thing in terms of the biology of the brain. Should we be studying it? What are some of the implications? What of these statistics is related to lifestyle? What of it is biology? We don't have the answers to that yet, and we currently do not have anyone studying those problems and or those questions. And there are a lot of related questions in terms of the neurochemistry, but I think the specific question you asked, we don't have anyone. It'd be great to get someone. I hope that over the next decade, we'll in general just really see a growth of interest in discovery in that field. Thanks.
Yeah, and I think I'll just follow up to what Daphne said with what we were talking about at dinner about hormones and not really understanding that side of it at all. As Daphne mentioned, we don't really understand that. But the one side project I guess the lab does have related to this, which is still in play. We haven't cracked it yet, but we are certainly working on it. Is that one of the most surprising statistics that I found, going back to Ken's idea of kind of looking at statistical correlations, is 1 in 3 women who have preeclampsia go on to develop vascular dementia. And vascular dementia is not quite Alzheimer's, but if I describe what it is, you'll kind of get the idea of why this might be relevant later in life. So when I was a postdoc in 2014 or whatever, and I was reading a paper about these amyloid fibrils, these things that are protein filaments that form aggregated proteins, usually found in neurodegeneration. Suddenly they were appearing in the urine of women who had preeclampsia. Totally different part of the body, totally different process, much younger. And what's thought is that whenever you have such high blood pressure as you do in preeclampsia, dangerously high blood pressure, sometimes fatal blood pressure, hypertension, it's putting so much pressure on the kidneys that they start to excrete through the wall of the kidney very high concentrations of protein. And at high concentrations of protein, that's when you start to get these amyloid fibrils. So that's also why, for example, people who have genetic predisposition to Alzheimer's get more plaques, because they just overexpress these particular aggregation-prone proteins. And so it's a bit simplistic, but one of the things that we're trying to figure out right now is if we can use cryo-EM to identify the structure of those aggregated proteins in women's urine, well, they are already in the bloodstream of that woman. And one thing that is known is that amyloid fibrils tend to kind of spawn other amyloid fibrils. So you can have an amyloid fibril made from A beta, and that can also cause the aggregation of tau, let's say. And so the thing with vascular dementia is that the fibrils are building up in the brain vessels, the capillaries of the brain. That's also where A beta is. And so one idea is that are those fibrils that appear in your 20s, 30s, maybe early 40s that continue to circulate around your brain, are they triggering A beta accumulation later in life of those women who had preeclampsia? And there is precedent for this because there is another condition where above the age of 60, I think, there's a heart protein called medin, and that forms amyloid fibrils, and that can also trigger A beta aggregation leading to Alzheimer's. So it's a thought. We haven't finished it yet, but we're working on it actively.
So another hot subject is the gut microbiome in the other brain, and I'm sure something's going on there. Maybe you want to expand on that?
I can talk about that. I think for George's question, I would refer you to Charles Zucker. Charles's new direction is all about exactly what you just described. I think they even have a company now as well, describing, what is the link between the gut microbiome and the brain, particularly through the vagal nerve. There do seem to be kind of patterns of certainly, the strongest evidence for that, I guess, is with alpha-synuclein. So we do find alpha-synuclein fibrils in the gut. There have been experiments where they snip the vagal nerve and then they don't see them appear in the brain of mice. And then if they, I don't know, there's a whole field about this that I am not going to speak about, but Zucker is actively working in this area, who's a member of the Zuckerman Institute, if you don't know. So do you want to?
No, that's everything to say, and there's a lot to say about it. It's also this lab, Charles Zucker's lab, also has some new studies about similar kind of brain-body interactions around inflammation and signals coming from the brain to regulate inflammation in the body and vice versa. I think that's another area we're going to see a lot of growth in the next several years. We're just starting to see these inkling of findings that are really, if anything, just proving how many questions we haven't even asked, let alone answered. Yeah. I think with all the talk about sleep and how important it is, and it's been an information-dense evening, maybe we'll stop there just to thank you all for joining us. We're around to answer any questions individually and to chat for those who would like to remain a little longer. And enjoy the rest of this beautiful weekend. And finally, to end by thanking Ken one more time for bringing us all together for this memorable evening.